Hi, I'm Paula Hammond. I'm a member of Old South Church, but I'm also a polymer chemist and a chemical engineer. I'm a professor at MIT in the Department of Chemical Engineering, and I'm here in my office at the Koch Institute for Integrative Cancer Research on MIT's campus. I'm really excited to tell you a little bit about the kinds of things that I do. In my lab, we design new materials that can encapsulate drugs and deliver them directly to certain parts of the body. We use a very simple method that involves large molecules known as polymers. These molecules are known for a number of different applications. You can find polymers that are part of the packaging that encapsulates our food, but you can also find polymers as naturally occurring molecules in our body. For example, proteins are a kind of polymer or macromolecule. In the work that we do, we design polymers that can encapsulate a range of different proteins and small molecule drugs. And we use this approach to create extremely thin coatings that can coat the surfaces of a range of different biomedical devices. This includes, for example, coating a biomedical implant with layers of polymer that contain a drug like a growth factor. The growth factor is something that can stimulate the generation of bone. And what we do is encapsulate the protein so that it is slowly delivered near the part of the body where we want new bone to grow. We also have projects in which we design layers that can release from extremely small nanoparticles. This is actually a part of our work in addressing cancer. In this work, we can take these extremely thin layers and we alternate the layers by charge because opposite charges attract. So we can take a negatively charged, very small particle and wrap it with a positively charged polymer. We can then put down another negatively charged material. And in our case, that negatively charged material is RNA. In our case, silencing RNA, a molecule that can silence genes in the body. The reason this is important is because a number of cancers are the result of the modification of genes, and silencing RNA can actually silence those genes, make those cancer cells very susceptible to the chemotherapy that we put into the core of our nanoparticle. Now I'm going to show you a very simple model of the kind of particle that we generate in our lab. Here we have a big particle, but in real life, we make these particles incredibly small. They are 100 nanometers, which means that they are just 1/1000th the width of a human hair. And that means that the particle can travel through the bloodstream. And then when it reaches the cancer, it can get caught up in the tumor and it can actually localize to tumor cells. So here's our nanoparticle, and it's designed so that its outside loves water and is very hydrated. And that allows the particle to go through the bloodstream. We attach ligands to the outside of the nanoparticle so that the nanoparticle can tune to the tumor. So these are small groups which attach to the outside of the nanoparticle and recognize a protein on the outside of the cancer cell. And that makes this a tumor homing nanoparticle. So this is the outside of the nanoparticle, but inside we have more layers. In our case, we actually contain an inner layer which has the siRNA or the silencing RNA that can turn off the genetic code in the cancer cell that allows that cancer cell to survive in the presence of chemotherapy drug. Once that siRNA is released to the cell, inside we actually have a drug molecule, and that drug molecule is contained within a liposome. So we can see this is another little carrier of drug molecule that gets released inside the cell. And then ultimately, our drug gets released. One of the exciting things about what we do in the lab is design materials to deliver drugs and address human disease. And this is really exciting because some of the work may translate to actually address patients. And this is one of the goals in our work. We're very interested in how we can advance this work so that the medicine can actually get to patients in a more effective way while lowering the side effects of chemotherapy. There is another aspect of the work that I do, though. I'm also a teacher. I have students in my research lab who are learning to become experts in the field. And I'm very excited about the fact that they are going to go on and complete their PhD degrees, having learned a large number of laboratory skills, but also skills in how to think and how to do research. I'm really proud of the fact that the students get a chance to go on and explore their futures after working in our lab. One of the reasons that I get involved in science is that as a child, I was very interested in everything that happened with the natural world. I was interested in how leaves came out of the trees. I was interested in ants and small animals. I was interested in how nature works. As I got older, I was also very interested in my science and math classes. And ultimately, I got excited in chemistry class when I got to mix chemicals together and see things change before my eyes. Maybe some of you are excited about science too. And I think if that's the case, that's wonderful. Whether you already have a job and you like to learn about science on the side, whether it's part of the core of what you do, or whether you're very young and you're thinking about what you might do in the future, I think that having a greater knowledge of science helps all of us as a group because we can learn how science can help advance the human problems. So it was great sharing some of the work that I do with you, and I'm looking forward to a chance to see you at Old South at some time in the future. Thank you.